Sciencera. In this video, we're going to discuss some key terms to remember before we start this unit. Immunology, a study of a host reaction when foreign substances are introduced into the body. As a science has its root in the study of immunity, the condition of being resistant to the infection. Antigen. Foreign substance that induce such as an immune response is the antigen. All immunogen are antigen, but not all antigen are the immunogens. Immunogens are antigen, macromolecules capable of triggering adaptive immune response by inducing the formation of antibodies. Immunity, condition of being resistant to infection. Let's start with the immune system definition. The immune system is structured to recognize and respond to and destroy a wide variety of invading organisms that would otherwise be capable of promoting infection harmful to body substance. Two types of immunity. First one is natural immunity, individual ability to resist infection by means of normally presenting body function, non-specific, naturally occurring at birth. Lack of memory cells, example skin and macrophages are the part of natural immunity. Natural killer cells are also part of natural immunity. The second type is acquired immunity. Resistance characterized by specificity for each individual pathogen, specific immunity which is not present at birth. It has memory B cells. Primary response first encounter the antigen, body meet antigen the first time around. Secondary response is when already meet first, second and so on. It is better and faster than primary response. It has diverse response in each antigen. Defense system. Physical and chemical defense system. Starting with physical barriers, composed of structural barrier that prevent most infectious agent from entering the body. It is the first line of defense in the immune system. The skin has thick layer of dead cells in the epidermis which provide a physical barrier, periodic shedding of the epidermis which remove microbes. The mucous membrane produces mucus that trap microbes. Hair within the nose filters air containing the microbes, dust and pollutants. Cilia lines the upper respiratory tract, trap and propels inhaled debris to the throat. Urine flushes microbe out of the urethra and defecation and vomiting expel microorganism. The next is chemical barriers. Designed to recognize molecules that are unique to infectious organism, it is second line of this defense. Phagocytosis, engulfment of cell or particulate matter by neutrophils, macrophage and other cells. Lysosome is an enzyme produced in tear, break down cell wall and thus act as an antibiotic. Gastric juice in the stomach destroy bacteria and it is highly acidic. Saliva dilutes the number of microorganisms. Acidity on skin inhibit bacterial growth. Sebum provide a protective film on the skin and inhibits growth. Hyaluronic acid is gelatinous substance that slow the spread of toxinous agent. Next is cellular humoral component. Cellular component phagocytes are the white blood cells, granulocytes, natural killer cells, kills virus and tumor cells prior to exposure include lymphocyte. Lymphokine activated killer cell or LAK is effective in killing virus, kill cancer or tumor cell. It has been shown that lymphocytes when exposed to interleukin 2 are capable of lysing fresh non-cultured cancer cell both primary and metastatic. The only problem when LAK cell uses higher interleukin 2 can have side effects such as severe fluid retention. Then there is humoral component, which include compound substance, cytokines, complement pathway, lysosomes, antimicrobial substance, and beta lysins. Cellular humoral immune system versus cell mediated immune system. In humoral immune system, A B cell binds to the antigen for which it is specific usually requires cooperation from the helper T cell. The B cell often with stimulation from the helper T cell differentiate into the plasma cell and then plasma cell proliferate produce antibodies against the antigen. Cell mediated immune system. A T cell binds to MHC antigen complexes on the surface of the infected cell activating the T cell with its cytokine receptor. Second, a helper T cell produces cytokines that cause the activated T cell to differentiate into a cytotoxic T cell. These cytokines also influence the formation of plasma cells and activate macrophages. Lastly, the infected target cell is lysed with cytotoxic T cell. Cellular defense mechanism. The first cell in the cellular defense mechanism is neutrophil, which is 50 to 70% in the circulating white blood cells. Primary granules contain myeloperoxidase, elastase, proteinase 3, and lysosome, cathepsin G, and defensins. Secondary granules include collagenase, lysosome, lactoferrin, NADPH, etc. And tertiary granules include glutinase, plasminogen activator, capable of process of diapedesis, adherence in the cell wall. Pseudopods are the phosphat present in neutrophils. Example chemotaxic factor. Neutrophil may wander randomly through the tissue, can be attracted to specific area. Chemotaxin is the chemical messenger cause migration cell. Chemotaxis process three stages. First stage is migration and adherence. Second stage include form of pseudopods through and squeeze in junction. And third, chemostaxis if have chemotactic factors. Next is eosinophil. 1 to 3% in circulating white blood cell, increase in allergic reaction and parasitic disease. Reddish orange granules also called Roman sky stain. Primary granules include ACP and aryl sulfatase. Secondary granule include major basic proteins, eosinophil cationic protein, eosinophil peroxidase, eosinophil derived neurotoxin, phospholipase, histaminase, aminopeptidase, and ribonuclease. ADCC process antibody dependent cellular toxicity is dependent on the antibody. Parasite, for example IgE, the granule will poke out and leave out. The toxic substance released by the cell are 1. Killing the micro basophil, less than 1% in circulating white blood cell. Bluish purple granules contain histamine, eosinophil chemotactic factor of anaphylaxis, heparin, involved in immediate hypersensitive reaction type 1. Mast cells can be found in connective tissues. Allergic reaction basophil always pair in mast cell. Granule contain ACP, LP, ALP and protease. Monocyte largest white blood cells in peripheral blood, 4 to 10% of circulating white blood cells. No megakaryocytes on peripheral blood. If visible, the bone marrow is affected. Possesses grayish-blue cytoplasm and is ground glass in appearance. Granule contain peroxidase, ACP, and aryl sulfatase. Other type of granule contain B-glucuronidase and lysozyme lipase. Precursor of cell of macrophages are monocytes. Example alveolar cell, lungs Kupffer cell, liver histiocytes, macrophages. Large version of monocytes on tissues are macrophages. There is increase in number of ER lysosome and bacteria, mitochondria. Granules contain no peroxidase at all compared with monocytes. Monocyte macrophage system function in microbial killing tumoricidal activity, killing of intracellular parasite, phagocytosis, secretion of cell mediators, and presentation. Dendritic cell involve APC antigen presenting cell present antigen to the T cell. Most potent function is to phagocytose antigen and present it to the T helper cell. Most potent phagocytic cell in tissue. Toll-like receptor protein discovered in fruit fly Drosophila like molecules found in human leukocyte. Antifungal immunity, highest concentration in leukocytes. It provides surveillance and invariant recognition mechanism that detects specific and unique molecular pattern associated with pathogen and inflammation. NBC-LLR proteins, nucleotide binding site and leucinuric repeat 21, more than 13 toll-like receptor. Immune system. Phagocytosis, engulfment of cell in particulate matter by leukocyte, macrophage and other cells. Chemotaxis, cells are attached to the site of inflammation by chemical substance. Positive chemotaxis near the site or towards the stimulus, and negative chemotaxis away from the site or away from the stimulus. For example, C5a, C5b, C6 and C7. Opsonization, substance that adhere to antigen to help the phagocyte during the process of phagocytosis, coating of antibody and or the complement to facilitate phagocytosis. For example, C3b, C4b, C5b, fibronectin leukotrienase, and immunoglobulin. Process involving phagocytosis. Physical contact between white blood cells and the foreign particles through opsonization or attachment, enhanced by opsonin. Formation of phagosomes. One attachment has occurred, the cellular cytoplasm overflows the particle to form phagosome. Fusion with cytoplasmic granules to form phagosome. Digestion and release of debris to the phagocytosis types. Indirect, via opsonin receptor that recognize opsonins such as IgG, CRP and C3b bound to microorganisms. Direct phagocytosis via the pattern recognition receptor toll-like that recognize lipid and carbohydrate sequence on microorganism. Pathogen killing. Oxygen dependent. Respiratory bursts happen on the second stage of phagosome occur when the cytoplasmic pseudopods enclose the particle within a vaccine. Hexose monophosphate shunt utilize NADP and NADPH product hydrochloride ion. The more continuous the process, the more toxic to microorganism. Oxygen independent. There is pathway happen product formation toxic substance such as nitric oxide, production of nitric oxide from oxidation of largine by nitrogen oxide synthase, which is produced by IFN gamma activated cells. Nitric oxide synthetase is induced when phagocytic cells meet organism. Nitric oxide, soluble, high labile, free radical gas that is capable of operating against organisms that invade the cytosol. In the presence of other reactive oxygen species within the phagosome, nitric oxide is converted to peroxynitride, another product which are highly toxic to bacteria, yeast, and viruses. This brings us to the end of today's video. Thank you for watching. If you like my video, don't forget to subscribe for more.